So, good afternoon, or good evening, or good morning, depending on where you are in this world. I want to give you a warm welcome at this presentation for the Master Life Science and Technology. My name is Hilde Zwaan. I'm study advisor for this program, and I give this presentation together with Professor Lars Jeuken. He will tell you more about his research and also about a course that he gives in this master. And also Matthijs will tell you something about his experience as a student with this master. In the final part of the presentation, I will give you some more information about the job prospects, and we will handle the questions from the chat. So please, if you have some questions, put them in the chat. But first, it doesn't. Okay. But first, the master. So this master is embedded within the Leiden Institute of Chemistry, just like the master chemistry. And this institute has two research themes. We have chemical biology. Here scientists want to understand biological processes in the cell at a molecular level to understand certain diseases, how they develop, how they interact. And they are looking for potential medicines or antibiotics or vaccines as a cure. And how we synthesize with organic chemistry, potential drugs are synthesized and also tested in cells. And we have also another research theme. We have energy and sustainability. This is to develop new materials and processes that are needed from the transition from fossil fuels to sustainable alternatives for both energy and also chemical products. So think about the conversion of CO2 with catalysts, or the splitting of water into hydrogen for more sustainable energy. So within the LST, we offer 4 specializations. We have the research specialization, if you are interested in doing a PhD, or if you want to do research at a company. We have the business specialization, if you want to do a more position in management after this master. We have science, communication, and society, if you are interested in learning how to communicate science to a broader audience, like product technology, health issues, environmental safety. You learn that here. And we have the education specialization, if you want to become a teacher at a high school. Here you see an overview of the 4 specializations and all the components, and you see that the first 2 rows are the same. So within each specialization, you do 4 core courses of 5 credits each. We also offer the component academic and professional skills. This is about academic writing, peer review, responsible use of AI, data management, and also career orientation. But then you see some differences. So for the research specialization, you do a minimum of 60 credits of research. And then for the other specializations, you do a minimum of 30 credits. So then you have the electives. I will tell you more about that later. And the specialization-specific components. For business, this is 40 credits. And this involves 20 credits of business courses in marketing or financial management and other courses. And you do a business internship, not in research, but at a company in a management position of 20 credits. We have also the science communication, and there you have a program of 40 credits, and you do 23 credits of courses in science communication, and you do a project of 17 credits within science communication at the university or outside. And then within the education specialization, depending on whether or not you have done the minor education program in your bachelor, you do a program of 30 or 60 credits. So about the electives, In this program, we offer a lot of elective courses. You can also do more core courses. The minimum is 4, but we offer 9 core courses. You can also do courses at the Faculty of Science of other master programs, also here in Leiden or at other universities. But there is also there are some other opportunities. So you can also extend the research component of your program. So with up to 20 credits, you can extend this component. And you have also the possibility for the business and the science communication to use up to 20 credits to extend this specialization. So you can do more business courses, business elective courses. You can increase your, extend your business internship, or you can extend your science communication project. And I always say that the specialization is an opportunity and not a limitation. I see people doing research and then after their graduation, they decide to pursue a career more in management, and they have developed their analytical skills, and they can use that outside research as well. And also the other way around, I see also students doing business, and then after their business internship, they compare the business internship with the research project, and they decide to pursue a career, or they decide to do a PhD position. Why would you study LST in Leiden? So Leiden is a very nice city with a very good atmosphere. And I've shown you that you have a lot of freedom. Yeah, you can choose your specialization. You can also extend your research component. You have a lot of choices in courses. And there's also flexibility. I see also students starting with, for instance, the business specialization. And then after a few months of doing courses, they like this so much that they switch to research. So that is also possible. You can do your major project here at the Leiden Institute of Chemistry, but you can also go to the Leiden University Medical Center or the Erasmus Medical Center or the Dutch Cancer Institute, or here at the Institute of Biology and Drug Research. And there's also a possibility to go outside these institutes, but I will tell you that about that more later. Also, the courses that we provide are linked to the research within the Leiden Institute of Chemistry. And I will show you this with the core courses. So we have 3 categories of core courses: biophysical sciences, molecular sciences, and biomedical sciences. And students must do 4 courses out of 9 courses that we provide. And you have to choose within each category 1 course. So for the biophysical science, we have these 3 core courses. And for instance, you see here the research of Professor Marcellus Ubbink, and he teaches the course Enzyme Dynamics, NMR Spectroscopy, and Kinetics. For molecular science, we have also these 3 core courses. And you see, for instance, the name of Professor Sylvestre Bonnet with his research, and he teaches the course Metals and Life. For biomedical sciences, we also offer 3 courses. You have to do 1 in each category. And here you see the name of Professor van Kasteren, and he teaches immunotherapy and technology. So we see and we hear from also external scientists that these core courses provide a good basis for research. And you, of course, you can also do more specialized courses as elective courses, but these core courses are the basis of this program. And we hear from, we have an external scientist, Laila Akari from the Dutch Cancer Institute, and someone from the Wayne State University, and they confirm this is, they have seen our students working in their lab for an external project, and they confirm that this is a good basis for research. So what I told you is that the major can be done, your major research projects can be done here at the Leiden Institute of Chemistry or at the Leiden University Medical Center, the Erasmus Medical Center, the Dutch Cancer Institute, or we have also the Institute for Biology or the Institute for Drug Research. And then you can also do another project externally. So for the research specialization, you can do an optional minor research project with a minimum of 20 credits up to 40 credits. When you use your electives, you can do more research. Within the business specialization, you do the business internship of 20 credits, but you can also use your electives to extend it to 30 credits. And again, this is not a research project, but this is really something what you do in management, in marketing, or in sales. And then we have the science communication specialization. And here you do a project of minimum 17 credits up to 37. So you can do it here, but you can also do it, for instance, at a magazine of New Scientist or at a museum like the Naturalis here in Leiden. So this was some general information. I will now give the word to Professor Lars Jeuge. So,
thank you, Hilde. So I'm Lars. I am one of the academic staff that teaches on the LST Master's program as well as in the Bachelor's program. And I'll say a bit on how the staff is involved in the Masters. So in Leiden, most staff are connected via different research groups where multiple staff are combined, usually also sharing lab spaces. So master students who do their project in those labs find themselves in a corridor where multiple staff members actually share that space and share their knowledge all the time. So I work within the macromolecular biochemistry labs. We actually have the same sort of viewpoint and vision as chemical biology. We try to understand biological processes at the molecular level. And there we focus, of course, as the name suggests, on macromolecules. We specifically focus on the function and structure of membrane proteins, as they have a very important sort of function in health and disease. And we also look at DNA and RNA and protein-protein interactions and protein folding. And you see here the names of the other staff that are in our group. So I would like to say a bit about one of the projects that runs in my lab by one of my PhD students. And masters sometimes join this PhD students in his project. So what you see here is the electron transport chain of Mycobacterium tuberculosis. As you might know, tuberculosis still kills about 1 million people worldwide each year, and 15 million people are infected. So it's still one of the major health concerns we have, as also identified by the World Health Organization. What you see here, I hope you recognize as an electron transport chain. It looks a bit like that of mitochondria, but there are also a couple of differences. So here you see 2 enzymes that you don't see in mitochondria. And the general idea is if we can inhibit those enzymes, we can fight tuberculosis infections. We actually work on both, but I'm going to show you a bit on this enzyme over here, cytochrome BD, which is a terminal oxidase, so it reduces oxygen to water and thereby completes the electron transport chain of tuberculosis. So we start often with these types of things with the structure. This can be a structure that's already known. In this case, we did solve one of the structures ourselves. We have one of the a state-of-the-art cryo-EM facility here in Leiden, where we can do single particle analysis. That means you purify the protein. You put it in on the micrographs of the cryo-EM microscopes. You then select about somewhere between 50 to 150,000 particles, sometimes more. And from that, you align all these to get really high resolution structures on the different orientations of the protein that you find yourself on that grid. Then you use general software that is state-of-the-art in the field to align or to sort of orientate all those 2D images to create a 3-dimensional structure of your protein. And here in red of the membrane protein, you see a detergent micelle, and in blue, you have the protein itself. And then ultimately, you have resolved to a certain resolution your membrane proteins that you study. Then we only have the structure. That doesn't tell you very much. You want to, of course, know the function, and the function also in the disease state. So in this case, how it relates to the infectious sort of behavior of the bacterium. And therefore, we, of course, need to do enzyme assays. We need to study these proteins with a whole plethora of biophysical techniques. Here we see a very, very basic activity assay of this enzyme into a reconstituted minimal respiratory chain, as we call it, where we've only taken 2 of these enzymes out of the bacterium. And then you compare that, in this particular case, this is just some data that we have measured in the last year. 2 of the same protein homologs out of 2 bacteria, the tuberculosis, and we've compared that with E. coli. Of course, if you then interested in maybe creating medicine or study, can we inhibit this protein? What will be the effect on infection and the process of this bacterium? You can do a inhibition screen. So in this particular case, we have used a robotics to screen about 10,000 compounds. So each of these lines here, most of them are absolutely white, which means there's no inhibition, but then you see some darker lines. And these darker lines means that one of these compounds inhibits actually the catalytic activity of the purified protein. You then need to sort of limit it down. So in this particular screen, we had 50 hits about, and then ultimately we dwindle this down to 1 most promising candidate. And then you go all the way back to growth curves of the tuberculosis. And we find that our compound, which is here, has the acronym 17, is actually inhibiting growth. In this case, blue means the bacteria hasn't grown, and purple means it still grows. So here you sort of see a project from beginning to end, going from structure to function to inhibition to health and disease. The courses we teach from the biochemistry group are various. Some of them are core courses and some of them are elective courses. I myself teach on cell membranes and membrane transport. So we specifically look at the function of lipids. There's many, many different lipids in nature. What are the physical chemical properties of these lipids and how does that lead to the specific function they have? And why is a lipid has such different behavior than, for instance, detergents? And how do we use that in research? Lipids are also very important nowadays in drug delivery. The lipid nanoparticles I hope you have heard about. So which lipids would you choose for lipid nanoparticles? How would you make one of these lipid nanoparticles for drug delivery, and what are the properties? And you need to understand both the chemistry as well as the biophysical properties of these lipids. Other people in the biochemistry group, they look at protein folding and misfolding, and this again is very important things for things like neurodegeneration. So what happens when we have protein misfolding, for instance, in our brain? What do we understand of protein amino acids sequence, and how does that lead to misfolding? Other people look at specifically at photosynthesis. So here we see a lot of people also interested in artificial photosynthesis taking courses like this, because it helps to understand natural photosynthesis if you want to make artificial photosynthesis. So this is all about membrane and protein function, and of course misfunction or misregulation. And then we have courses on protein structure and dynamics. These 2 are both in the core, and they've already been mentioned. People of the biochemistry group teach in a specific course all about protein structure using the 4 main or the 3 main techniques nowadays for resolving protein structures. And then we have one that is more focused on enzyme dynamics, which has also the force method for protein structure determination, NMR, in them. So with these 2 courses, you basically have a very broad and complete in detail understanding of protein structural biochemistry. So with that, I would like to give the word to Matthijs, one of our students.
Yes, thank you very much. So Nitya, my name is Matthijs, and I've been a student for a little over 2 years now here. And I'm just going to tell you a bit about my experience as a student at the master program. So yeah, currently I am in my third year. I'm almost done, so now I'm finishing up with my colloquium, which is a review on literature on a certain topic that I'm doing. Just some background about me. I'm currently 23 years old. I'm an active member of LIFE, which is the study association of life science and technology. And up until very recently, I was the ambassador for the life science and technology bachelor, which is the bachelor that I've also followed, which is taught here also in Leiden, but in combination with modules at the Technical University of Delft. So my hobbies include reading, making music, and going on hikes. And my interests specifically for the field are mainly immunology and also the tumor microenvironment. So yeah, first of all, why did I pick this master program? Well, first of all, I of course had a passion for fundamental research in mainly the biomedical sciences. And I saw that there were really a lot of electives, both inside and outside of the Leiden Institute of Chemistry, which gave me plenty of room to sort of pursue my specific interests, which is something that really spoke to me and which I really liked. And having done the bachelor here, I already knew the institute and the people. And from my experiences, the Leiden Institute of Chemistry and the program really consists of quite a tight-knit community of students and researchers and professors, which is also something that I quite liked. So yeah, just an overview of my program. Of course, I had to choose 4 core courses, which in my case were chemical immunology, enzyme dynamics, chemical biology, and also biosynthetic and pharmaceutical chemistry. And you can see them here on the right. And then I actually also followed 2 other courses that could be a core course, but I took them as electives, which were metals and life, and also in vivo biomolecular interactions underlying disease. Yes, and then apart from that, I also obviously took my elective courses. And as you can see here on the figure of the right, you have to take 24 credits, but I was quite interested in the elective courses, so I took a couple more. And I actually took 34 credits of elective courses. So some courses included medicinal chemistry and drug discovery, and also cell membranes and membrane transport, which is the course that Lars teaches. And I also took molecular aspects of RNA viruses and a course on antibiotics, and also a course on laboratory animal science, which is a program that's actually taught in the hospital in the LUMC, which allowed me to get my certificate to do animal experiments. So if we go over back to September 2023 and sort of look at the timeline that was my master's. In my first year, I mostly just took my modules and my courses. So in the first 3 quarters of the year, I took core courses and elective courses. And then in the last quarter, I also did this laboratory animal science module that also included some practicals. And then in my second year, I basically spent all year doing my internship. So my master end project, which I did at the chemical biology and immunology group. And now that I finished that, I am doing, as I said when I started, my colloquium, which is a literature review. And hopefully, when I'm done in November-ish, I'll graduate and then go on to pursue my PhD. So just a bit of extra info about what I did for my internship. As I said before, I did it at the chemical biology and immunology group. And what I was mainly working on was the development of an in vivo assay to measure nutrient partitioning in the tumor microenvironment. So just to sort of briefly explain what that means is, well, if we look at a tumor like this, as you might know, and you can also see, it's not at all a very homogeneous tumor. And in fact, most tumors have lots of other cells in them except for just tumor cells. So there's immune cells, also sometimes cancer-associated fibroblasts, and all of these cells need food. And because in a tumor there's usually not a lot of nutrients available, these cells quite often have to compete for the amount of nutrients to meet sort of their energy demands. And the big question here is that we want to know which cells are eating and also how much they're eating. So what I was working on was to develop assays and probes that can measure uptake of, for example, glucose or glutamine. So you can see some of the probes that we're using here. And the idea here is that you feed these probes to the cells that you're interested in, and then they will take them up. And then in a later stage, you can actually click them via a method that's called click chemistry. And I won't go into too much detail about that. But the nice thing is that then we can visualize these probes using a flow cytometry, and you can get these nice looking histograms, and you can actually see how much of a certain probe or nutrient a cell is eating. So yeah, that's something that I was working on, and I actually really enjoyed that. And I also learned a lot of valuable skills. So I did a lot of flow cytometry, I worked with primary cell culture, and I got to do some animal experiments. And then I also, learned how to analyze my data. I worked on my presentation skills. And of course, I had to write a thesis. And we were also starting to write the manuscript for hopefully a publication. And I actually enjoyed it so much that this also inspired me to apply for a PhD, because I really liked doing the research. And I ended up getting a job in Glasgow in Scotland, where I'm going to start in December. So that's also a really exciting prospect. And I think I'm going to give the word back to Hilde now to finish the presentation.
Yeah, thank you. Thank you, Matthijs. Thank you, Lars. So as you have seen, there are many options, and we want to prepare you for the next step after your graduation. And we want to prepare you for that. We have an intensive course, academic and professional skills, and this also includes a component career orientation. In this component, you do some workshops from the Science Career Service department. We have many choices, for instance, a LinkedIn workshop. We have alumni events. The Study Association organizes these events, but also we as a program, we organize these events twice a year. There's also a job market here annually, a science career event, and this is all a part of the career orientation. And I also would like to emphasize that we have a strong community. We think it's very important to have a good onboarding program and to build on the cohesions between students. We have a student mentor group program, and then you also discuss the choices that you have to make. And also, if you are in doubt, you can also always come to me and make an appointment to discuss some options. Also, we have the master committee. Every year we have a new committee, and they organize social events, but also a career event to PhD or not to PhD. So talking about PhD, nearly 50% will do a PhD after their graduation. And but there are also some other job prospects, and I want to give you some examples. So here we have Niels, and Niels did a double specialization, research and business, and because of his business specialization, he did a business internship at BaseClear, and after that he worked for DAP Bio. And now he works at Delft Circuits. So this is a different path, because he's now working within sales. So different from the research. This is Wala, and she did her Bachelor Pharmaceutical Sciences in Utrecht. She did the research specialization, and she did, besides her major projects at the Leiden University Medical Center, she did external minor at the University of California in Berkeley. And after that, she did various jobs. She works at Immuntune and Symphonix, and she now works for ProQR within research. So, and this is Mireille. She did her Bachelor Molecular Biotechnology in Barcelona. and she did business specialization. So after her research project at the Leiden Institute of Chemistry, she did a project, a business internship at RespiQ. And after her graduation, she became a marketing manager at Isogen. So these are some examples of what you can do besides doing a PhD. If you have more questions, please send us an email. This is our mail address. We also have the website, so you can here find some information about admission requirements, application deadlines, but you can also chat with one of our students, with Lily. She's the student ambassador. And also, if you are really interested in this master, please come next year on February 6th on the on-campus Master Open Day. You are very welcome to come. And we will now go to the questions in the chat. I see some questions coming in. I think the first one was from was about, are there any other bachelor degrees besides LST that would allow you to enter this master program, if necessary, with a pre-master program? Well, we don't have a pre-master program. As you have seen in the, I will also stop the slides. So as you have seen in my slides, I gave you some examples from alumni. They also had a different background from life science and technology. Mireille and Wawad, they did biopharmaceutical science, and the other, I forgot. But we see students coming in from biomedical sciences or another program. What is important is if you go to the website, you need to make, for the admission, a placement letter, and you have to show that your program, your bachelor program, has about 100 credits of overlap with our bachelor program, the joint master that Matthijs did between Leiden and Delft. So look at the courses and find the overlap. But we see that also students from other programs, they join this master, and our admission, board of admission, they also see if the grades are high enough about the research that you have done. If it's still possible in your bachelor program to do a research project more in life sciences, I would like to advise you to do so if you are interested in that. I'm not sure if you have something to add.
Not on that question.
Okay, but this is okay. We can go to the next question. Which hard or soft skills? Which hard or soft skills can you acquire with this master? And how does the program guide personal development? Well, the development of soft skills are very important, and we also, for instance, for the career orientation component, you start in September with an assignment, with a reflection assignment. Where are you now in terms of how did you develop yourself in the master? What kind of skills have you developed, also soft skills? And what kind of skills would you like to develop now in the program? And also you will get some feedback. You make some kind of a reflection. And also my colleague from the Science Career Service Department gives you feedback, and she says, oh, think about this, or you can go to that event, or she gives you some advice. And I think that many soft skills are also interwoven within the courses, presentation, collaboration, perhaps.
I can add some stuff if you want. Yeah, so first of all, obviously you learn a lot of hard skills during your actual modules, and for me mainly during my internship. So I just learned how to do a bunch of techniques, how to do like cell culturing and flow cytometry, etc. And then there's of course also the component where you have to do academic writing, because you have to write your thesis, you have to present your work at group meetings, but also for a broader audience of people who might not necessarily know that much about your project. So you have to sort of be able to talk about your research with a more general public. And then something that you also have to do during the year is these work assignments, which are also, I think, really nice for developing soft skills. So I think you have to do 6 in total, and there's 2 that you have to do. And then for others, you can basically choose. So you can look at what skills do I want to develop, and you can go to these sort of small modules and follow those. So for example, you can look at how to set up a LinkedIn profile, or you can do more stuff about whether or not you want to do a PhD or not, like they talk about. And there's really a lot of opportunities for you to sort of grow in that sense. And then towards the end of the master, you have to do this reflection that you did at the start again, and also sort of look back at, oh, hey, what did I learn in the last 2 years? And where do I want to go from now? And then that's also read by someone, and they'll give you feedback on where you might want to go, stuff that you want to look at. So I would say that I was guided really well, actually, in sort of my choice of what I want to do after the masters and also during the masters. So yeah, I would be quite positive about that.
Yeah, I can add a tiny bit also on the courses that you've seen. So you might have seen that also in your undergraduate and your bachelor's already. So a lot of the courses on the master level are done with soft skills in mind. So we don't just have the classical lectures anymore. It's much more self-taught learning, where you maybe read let's say a real article, and then afterwards have to evaluate the quality of that article. You have in teams, you need to prepare a presentation for a case study. How are you going to solve things? So all those soft skills come back even in the master courses, which you have to do depending on the stream, up to 60 credits. So you see it in all levels of the masters. These soft skills are coming back.
Okay. We have also a question. Is it possible to apply for this program with a biomedical engineering bachelor? Yeah, what I said before, I would recommend you to make a placement letter, and to it is possible, and the Board of Admission will take it into account. They will also look at the total picture and also the research that you've done and your grades, of course. And then I see also a question: what is, in your opinion, the main difference between LST and biomedical science? And that I would like to broaden this question, because we often receive this question. Also, what is the difference between LST and, for instance, biopharmaceutical sciences or chemistry? And I would like to ask Lars to answer that question.
So there's definitely overlap. So in the life science and technology one, we focus more on the cell level and the macromolecular level. So we go all the way down to proteins. And then, for instance, Matthijs here looks more at the whole cell level. While at the biopharmaceutical sciences, you go more to the whole organism. So you look more really on the medical application. So you do pharmacokinetics, pharmacodynamics. I'm not saying we don't do that at all, but we look more on the biochemistry and the cell level. So it's almost like an overlaying Venn diagram. And we often see that a lot of our courses, for instance, are taken by master students who actually take the biopharmaceutical sciences as their masters. So there's definitely some overlap. The biomedical science master depends a bit on where you would go for the biomedical science master, because different universities have a slightly different take on that. Some of the biomedical science masters are more like the biopharmaceutical masters I've just mentioned. Others, the biomedical science masters, are much more human physiology. So you're really looking at how does a human as an organism function. So you go to the whole organism level again, but then specific on the human. So I hope that answers your question. Feel free to ask for more explanation via the email that was provided earlier.
Okay, thank you. Another question is, are there options to start a master earlier than September, for example, in February? Yes, this is possible. So a large group often starts in September, and also a smaller group will start in February. So we see that, and we see also that they do that without any problems. So this is certainly possible. We have still some time. I have also one frequently asked question. I'm not sure if people are watching, who do a mixed program, more life science and technology, more a broad program like we have from Leiden and Delft, and they are in doubt whether to what kind of site they want to choose. Matthijs, can you, because we have this joint degree, can you say something about your choice between the choice of Leiden and Delft, which is more the technology?
Yes, absolutely. So I did, of course, do a joint degree between Technical University of Delft and here in Leiden. And well, Delft, as the name says, is a technical university, and they also offer an LSD masters. But well, it's much more focused on the biotechnology side of things. So what you're mainly looking at is cells that are not human, but for example, you're looking at microbes such as yeasts or bacteria or other stuff, and you're looking at processes. So, for example, you're going to look at how can we have E. coli or yeast that make these products and really scale up this process to big factories, and they focus a lot on sustainability and the energy transition. So that's very applied, very technical, and there's that. And then in Leiden, it's much more, well, medically. So you're looking a lot more at just human cells, the human body, what's going on there. And it's also more fundamental research. So you're looking at stuff that we do not know yet, instead of taking stuff that we do know and applying them to society, which is more Delft. And I just found myself always being more attracted to sort of discovering new stuff and things that we didn't know yet, which automatically led me to Leiden. And also the medical context I found very interesting. So if you are having similar doubts, I would suggest to just take a really good look at what courses or modules that you're following now, which you really enjoy, and sort of start to look at which electives and which core courses you can take here, and if that sort of aligns with your interests. Because in the end, the courses is like they are the things that you're going to be doing for 2 years. So yeah, try and sort of look at which route you would be more likely or more willing to go on. So more of maybe fundamental medical research or more towards an applied biotechnology course.
Yeah, thank
That's
you.
what I would say.
Yeah, and if you want to have more detailed information about the courses, you can also go to our study guide, to our prospectus at the website. So I would like to thank you for watching. I would really like to invite you for our on-campus Master Day next year in February. I also would like to thank Lars and Matthijs. Oh, one more question. Oh, sorry.
Yeah, that only just came in.
How long are courses valid in case I need to work during my studies and need longer to finish my degree? Yeah, they are. If you stay enrolled in this program, the courses remain valid. We see that more often that students take longer than 2 years to finish their program. So this is not an issue. Okay.
I think the courses are always valid, right?
Yeah.
But if you take really, really long, then the master itself evolves, and then that's
Yeah.
But then we're talking many, many years.
Yeah. Okay. So well, thank you for watching, and have a nice day. Thank you.
Thank you.
Goodbye.
Thank you.